So next we have James Shine from University of Sydney.
All right. Hi, everyone. So I'm going to start off my talk today with a little video. Look at this guy go. I just can't believe this. I found this on the internet a couple of years ago when I was looking up skilled performance and sort of playing around, sort of watching these videos of people doing those fun things, balancing lots of different stuff or, doing some high-level skill. This guy just blows my mind. Like, I'm not a big hockey guy, but look at this. He's balancing on this board back and forth. He's got this, stick in his hands with his gloves on. He's keeping the puck in front of him. It's called deking, apparently, if I asked some Canadians about that. And he's got his eyes focused. He's even got a fan on in the background. And I think right now, if you think about the state of neuroscience, I don't think we've got the foggiest clue how on earth he does this, right? What we might do if we go around the OHBM and ask them where, he could, how we could sort of combine the functions of all this little boy's brain, we'd probably go and look in the cerebral cortex. So we'd probably say something like, all right, he's got his boots on, so we can sort of feel his boots, maybe in S1. Maybe he's holding the stick with his M1 or maybe, premotor areas of his cortex. Maybe V1 is looking at the goal, that he's trying to sort of focus his eyes on. Maybe he's hearing the sound of the stick smacking against the ball back and forth. That's in auditory cortex. And then maybe we'd imagine something, the context. Maybe, he's got the kind of ice hockey rink in front of him or something like that, and he feels the context of the sort of higher order sort of context he's in his medial temporal lobe. And maybe you could think of something a bit more abstract, like the goal, that he wants to keep his eyes focused and he wants to keep the stick, and the puck together. He's got this sort of abstract goal, maybe in his prefrontal cortex. And maybe there's even something a bit more nebulous we don't even know how to talk about yet, like his sort of sense of the feeling of his body in the space that he's in, this idea of a kind of bodily awareness in his parietal lobe. But the problem is, let's say you list all those different functions, you're then sort of stuck at that point. You haven't really got a next good question to ask, because how do these things all work together? How do we kind of combine them into a satisfying story about how the system actually works? So we're in a Cerebellar session, so what I'm going to do is rag on the cortex a little bit. God, it's beautiful. There's a study that's just come out from the Allen and Stanford called Microns, where you can go in and look at this little cubic millimeter of mouse visual cortex. And they've gone through and they've painstakingly electron microscopy and analyzed this little chunk of cortical tissue. And in amongst all of the blood vessels and glia, what you find is just an absolutely dense thicket of cells. The cerebral cortex is, to first approximation, just a bunch of cells connected together, and everything looks like it connects to everything else. There seems to be very little set of rules about whether or not you should not connect to something else. In fact, the rule is for connection. This has actually been, I like to think of this a little bit like spaghetti. Everything's connected together. It's very hard to find order. I'm not the first to point out this observation. In fact, one of my favorite neuroscientists of all time, Valentino Breitenberg, said this many years ago. He had a lovely set of works. He was really a theoretical neuroanatomist back in the day. So what he would do is he would look at the brain and he would think carefully about how the brain could be built, how you could organize it, what are the kind of rules that would go into building the brain the way that it's built. And he tried to think about whether or not some of the explanations we had about the functions that came out fit with the way that it's actually built. And I think if you ponder these things, it's a bit navel-gazy sometimes, but you can actually kind of come to the conclusion that some of the ways, the functions that we map onto the brain actually just don't make a mechanistic match to the way the brain actually is. So here's what Valentino said about the cerebral cortex. He said, the idea which fits the cortical network most admirably is that of an associative memory. This is, in a way, disappointing, since many of us would have preferred to think of the cortex as a great thinking machine, performing within itself the most sophisticated neuronal computation. If the cortex is mainly memory, the act of thinking must involve it in concert with other parts of the brain, and we are now called upon to unravel this more complicated operation. These were wise words said back in 1998. He's so wise, I'm going to give him a wizard hat, because I think he was able to see this big problem in front of us that I think we still haven't really grappled with as a community. Right? Here's this beautiful associative memory network that, if you take it to its logical conclusion, can do lots of really beautiful things, but there's lots of stuff that it's really, really bad at. Like, and I would argue, this beautiful thing that we saw in front of our little hockey player, coordinating complex movement. So the question is, if we're not going to look in the cerebral cortex, where should we look? No one's going to be surprised that I'm saying that we should go and look in the cerebellum. Over half the neurons in the brain, but wired in a completely different way to the cerebral cortex. So when you're up in the cerebral cortex, as I mentioned before, there's very little discerning connectivity. If you're next to another neuron, odds are you're going to make a connection in a synapse with them. In the cerebellum, it's really, really different. Cells have got a very particular target, and they'll go right past different neurons and not make any connections whatsoever, and then make really, really dense connections with a different population. So if we come back to our wizard Breitenberg, he said in the cerebellar cortex, now this is different from the cerebral cortex, there is not one positive reentrant loop at all in the cerebellar cortex. Right? This is the exact opposite of the cerebral cortex. All feedback circuits there involve inhibitory synapses. So it's a really, really differently organized topology. The interactions of different inputs are strongly dependent on their distance and relative position. So as I said, it's not just about being next to someone. You've really got to make sure you go in and target the particular neuron you're in the circuit. And the dendritic neighbors of trees of neighboring Purkinje cells are neatly separated, each keeping out the territory of its neighbors with hardly any overlap. Now, this itself is an absolute marvel to see, and if you ever get a chance to look at cerebellar anatomy, it's just beautiful and exquisite. There's these really, really strange lines all over the place. These neurons are projected in this direction, and then completely orthogonal to them, you'll find another set of populations of cells that are connected that don't actually overlap in any way whatsoever. And for those of you who like biology, finding a right angle in biology is really, really hard to do, right? Biology loves to do little blends of things together, but it really rarely finds things at a complete right angle. But that's exactly what we find when we look in the cerebellar cortex. So this is a diagram from a paper that a collaborator of mine now who used to be a postdoc in my lab, Eli Muller, and I have been working on, where we try to take very seriously the topology of the cerebellar cortex and its connection with the cerebral cortex and the cortex and the rest of the brain, and then think very carefully about what kinds of algorithmic instantiations that anatomy could have. So here what you're seeing is a little picture of the pontine nuclei in purple. They're the nuclei in the bulge of your brainstem in your pons. They receive the main output of the cerebral cortex. They then project into an area called the granule cells. These are the 50% of the neurons in the brain, really, really teeny tiny cells. They then have these little fibers, the green fibers, mossy fibers that come up and make these parallel fibers that spread out where they then make contact with the Purkinje neurons. There's that 90-degree angle I talked about. The Purkinje neurons sit basically at 90 degrees to these parallel fibers. They head down into the cerebellar nuclei, and then they leave back by the thalamus back to the cortex. So you saw some of that really beautiful work from Jun before, talking about those cerebellar nuclei. So through a series of worked examples and some connections with nonlinear control theory, we argued that you can think about this circuit as performing something like an expansion, a linearization of the signal, and a compression. So what do I mean by that? The expansion is pretty simple to think about. The granule cell population is just ginormous, right? 50% of the neurons in the brain. So as you project down a particular motor plan, let's say I want to move my arm side to side, or if you like the hockey example, you can imagine the little boy with a little stick. You can imagine that as that stick motor neuron comes down to the spinal cord, it's going to send a little projection into the pons, and it's going to spread it out to lots and lots of different granule cells. Because there's so many of them, there's heaps and heaps of different ways that you can map that different input, and you can essentially blend them together with other types of motor patterns that have come in. So if you're interested in this, there's a couple of really beautiful papers on this. A theoretical paper from Alex Kaika-Gajic and Silva in 2019 in Neuron, where they argue that this dimensionality expansion actually allows you to find separation between otherwise overlapping signals. And then there's been a really beautiful addition last year in Nature Neuroscience. Mark Wagner, Missinelli, showed that essentially the compression that you see actually gives you a dimensionality expansion that actually captures the major signals of variance in the data. So that's the idea. You've got an expansion that starts out. What about the linearization? This is a little bit more complicated, and as someone that doesn't have a background in physics or engineering, a lot of this is based on the work of collaborators Steve Brunton and Eli's insights. But the really beautiful idea here is that if you expand a signal out into this massively high-dimensional space, you actually give yourself a really good chance of finding really nice linear readouts of this now nonlinear space. So the idea is something like, let's say I want to find a nonlinear match between the input that I'm doing and some output. It's really, really hard for me to do that in a linear system. So what I do is I map it into this high-dimensional space, which is itself nonlinear, and now I learn a linear mapping of that space. So I've now turned the problem from a nonlinear one into a linear one. Turns out that that's a really, really effective way to actually map dynamical systems over time. But if you're interested, there's way more in the paper and in this source work. Once we've linearized our signal, now what we've got to do is compress it back so we can do something with it. And this is where those deep cerebellar nuclei that Jun talked about come in. Because there's so many fewer of these cerebellar nuclei, now you've taken this massively linearized signal, you compress it back down, and you feed that back to the cerebral cortex via the thalamus. And now you've got the ability to actually take that information and do something with it. You can use that to actually anticipate what will happen next. And there's some extra cool stuff that the cerebellum does that I always love to throw in. The cerebellum on the way out, rather than just being inefficient, it actually sends projections back up to the cerebellar cortex, where it can actually learn to condition on sequences. And so there's some beautiful work from Michael Mork's group at Texas that actually shows that you can condition sequences of essentially responses to individual inputs in a rabbit, but via its cerebellar circuitry. So that's a lot of complicated concepts. I want to give you an intuition about why we think this is really interesting. And the way I think it's really helpful to think about this is imagining what your brain state's doing over time. So imagine that this little boy's got his little hockey stick and he's trying to balance. He's got this incredible, difficult problem. He's trying to parallel process his eyes, his ears, his hands, his feet, all at the same time. And we're trying to imagine that as a ball rolling along a landscape. And if you imagine all those balls rolling together, it'd be very, very hard to keep them on track. And so what we imagine the cerebellum doing is creating little rivets in this attractor landscape so that you can now allow the ball to roll in a really reproducible way, in a ballistic way, that allows you to anticipate what will happen next. We can quantify that with a bunch of different mathematical approaches. The one that we really like and that we've been playing with is basically look at the states that the brain can be in, map them all out, and then ask how likely it was that you were to see a change the way that you saw it. And the more unlikely something was, the less energy it would take. So the intuition here is that you've got this system. Every time you make a movement, you essentially project a signal down into your pons. You spread that signal out into your cerebellum. You then get a feedback that then gives you a particular state that you'd move into. And then you can move back and forth across these states, creating a sequence, which is essentially giving rise to the boy playing with his little hockey stick. So this has all just been a story so far. Is there evidence for it? So fortunately, there have been these really, really beautiful studies. We're fortunate to collaborate with Nuo Lee in the US, where they've done these really great experiments where they'll actually go into a mouse that's performing a sensory-dependent experiment where they actually have to hold an item over a delay and then respond to get a reward. And then they can go in and actually optogenetically stop their cerebellar nuclei from projecting back to the cortex. It can essentially obliterate the cognitive performance. And what Eli can then do is he can go and look at the actual activity that he sees in the data, and he can show that it actually becomes far less linear when you inhibit the cerebellar nuclei, and it actually becomes less compressed, and you make more mistakes. So these kinds of signatures that we expect to see in the data that come from an algorithmic exploration of the actual circuit are actually predictive of the performance of the animal. All right, I've gone a little bit over time, so let me just gesture towards some stuff that we think is really cool here. People have obviously been talking about the idea of the cerebellum expanding over phylogeny. I think this actually has a really, really important role for us to think about how cognitive performance could actually work in the light of these circuits. So Eli looked at some parallel processing data while people performed essentially a balance task and a maths task at the same time, and you essentially found that the cerebellum was incredibly important for performing that task. And Josh Tan, who's a really talented grad student who's been working in my lab, is about to move over to Korea, essentially has been looking at really interesting cognitive tasks like sensory motor adaptation and working memory manipulation tasks, among others. And you essentially find that the corticocerebellar system is really important for all these extremely cognitive tasks. But importantly, when you make mistakes, you actually see a slip off of this attractor landscape. You become much more variable on the trials where the performance was deficient. So I think there's much more to be said here. This is just a cerebellar-centric perspective, but I'm really interested in trying to take this into other areas like the basal ganglia and the superior colliculus. Please stop me if you'd like to chat about this kind of stuff. But in summary, I think as a field, we've been really, really corticocentric, thinking about the mechanisms that could give rise to behaviors like motor performance, but also things like cognition. But by opening up our eyes to the rest of the system and really thinking carefully about how the system can actually communicate together, we can come up with what I think are more interesting testable predictions about how the actual system can support complex behavior. And with that, I'd like to say thank you particularly to Eli and Josh for doing all the hard work. Thank you.